Good day, this is the Professopher, CBMD PhD, and today is Thursday, December 8th, 2022. It is 3:22 and 52 seconds, a.m. Eastern Standard Time. And today's research, cancer genes. So let's hop to it here. By flipping the switch, scientists shed new light on genetic changes that turn on cancer genes. New research on cancer causing genetic mutations may lead to improved ways of predicting and treating the disease. Date December 7th, 2022. Source: Salk Institute. Researchers have zeroed in on specific mechanisms that activate oncogenes, which are altered genes that can cause normal cells to become cancer cells. And this work advances the ability to predict and interpret which genetic mutations found in cancer genomes are causing the disease. As cancer can be caused by genetic mutations, yet the impact of specific types, such as structural variants that break and rejoin DNA, can vary widely. Researchers show that the activity of those mutations depends on the distance between a particular gene and the sequences that regulate the gene, as well as on the level of activity of the regulatory sequences involved. And this work advances the ability to predict and interpret which genetic mutations found in cancer genomes are causing the disease. If we can better understand why a person has cancer and what particular genetic mutations are driving it, we can better assess risk and pursue new treatments. Most genetic mutations have no impact on a cancer, and the molecular incidents that lead to oncogene activation are relatively rare. Laboratory studies how genomes are organized in 3D space and seeks to understand why these changes happen in some, but not the majority, of circumstances. The team also wants to identify factors that might distinguish where and when these events occur. A gene is like a light, and what regulates it are like light switches. They are seeing that because of structural variants in cancer genomes, there are lots of switches that can potentially turn on a particular gene. Using CRISPR-Cas9 gene editing, the research team introduced genetic mutations by cutting DNA in certain locations of the genome. They found that some of the variants they created had major impacts on the expression of nearby genes and could ultimately cause cancer, but that most had essentially no impact. And some genes appeared to go haywire when they were brought into environments with novel regulatory sequences, and others were not affected at all. The type of sequence that was introduced appeared to have a huge impact on whether or not the cell became cancerous. The next move is to test whether there are other factors in the genome that contribute to the activation of oncogenes. The research team are also excited about the new CRISPR genome editing technology they are developing to make this type of genome engineering work much more efficient. And this is the Professor of Microbiology, Bioinformatics, and Pathophysiology. This video was created for educational purposes only. Hope you enjoyed listening. Until next time, have a great day.